the principal organizer. Arpita will be the moderator today. I just want to thank everybody for accommodating us in this rather tumultuous time. And we look forward to hearing talks today and having productive discussion today, as well as Friday. And the information is in the email. And if you have any questions or comments or issues, you can reach out to me. You can reach out to the PoLS leadership. But thanks for joining us today. And I'll be quiet.
Okay. Good morning, everyone. I'm Arpita Upadhyay. Faculty.
He's a Georgia Tech, very Southern fellow.
Oops, sorry. At the University of Maryland in College Park. Thank you all for joining today. I'm happy to welcome you to today's session, which, as you've heard from the emails, will be a shorter than planned session. I just want to say I do support everyone who's decided to speak either today or on Friday, and we will have a longer, more focused discussion on Friday on this topic of equity and diversity in STEM and academia and physics of living systems. So with that, I just want to point out again, in case there's some newer people, today we are running this as a regular Zoom meeting, and the talks will be a total of 15 minutes with 12 minutes for the talk and 3 minutes for questions. So whoever wants to ask a question, you can either raise your hand using the raise your hand feature, which is at the bottom of the participants list.
No, we don't,
right.
It's not a webinar anymore, so there's no raising hands. So if you want to ask a question, you can just talk.
Okay, there is a raise a hand feature on the bottom.
Oh, really?
Of the
okay, I can't see it.
Yeah.
Sorry.
Yeah, because you're the host. So,
yes.
And Margie, I think you should make me the co-host so I can see who raises
Sure.
their hand. I'm not sure if I can see it otherwise. But you can either raise your hand and ask your question by speaking out loud. Or you can ask a question using the chat, because I do not see a Q&A over here. So you can ask a question using the chat and I will ask it to the speakers. Okay, with that, I'm happy to introduce our first speaker, who is Fernando Bruno da Silva from Universidade Estadual Paulista in Brazil. And he'll be talking about non-native interactions modulate protein folding kinetics. And Fernando, you can, when you're ready, you can go ahead and start sharing your screen. Okay. And take it away. Yeah.
Okay, I'm gonna share my screen. Okay, can you see my screen and can you hear me well?
Yes.
Okay, thank you, Arpita, for the introduction. First of all, I would like to thank you all the organizers for the invitation. It's really a pleasure to be here to present my results. So my name is Fernando Bruno. I'm a graduate student in molecular biophysics at São Paulo State University. One of the talks that I'm studying with Professor Victor Leite is how non-native interactions modulate protein folding kinetics. And today I'm going to show you some results that I got in these last years addressing non-native potentials into a structure-based alpha model. Here we have the spectrum problem that was reported before by Jenny Clark and Ben Schuller group. And the most interesting thing here is we have three domains for out spectrum that you can observe. We have almost no different structure here. And the question that involves this domain is, why does the domain R15 folding with 3 order magnitude faster than its homologs? Basically, the key answer to this question was given by these two groups, and the internal friction is responsible for this effect. But what is the internal friction here? We can understand that internal friction is a term to describe a collection of non-understood behaviors. And because of this, we try to understand the internal friction in terms of non-native interactions. So here I'm going to show you our results that I got when I performed the simulation with just use the structure-based alpha model as no vanilla model. And here we have the ratio folding time to domain 16 and 17. And as you can observe, you have almost no difference between these values. But if I have a non-naive interaction into a vanilla model here, in this case, we have the HAP that I call hydrophobic effect is given by a mesoid joining potential. And here you can see that our folding time to protein 17 increased a little bit. At the same time, to the protein 16 remain almost the same. But when I change this non-naive interaction to electrostatic interaction that is by the Hooke potential here into a vanilla model, we can see that, sorry, we can observe that I call a behavior swap here, because if you just take a look to the protein R17, the folding time, its folding time decrease at the same way the folding time of protein 16 increase a little bit. Because of these different behaviors, I'll try to understand if I have this two non-naive interaction into a vanilla model, I will have a cooperative or a non-cooperative effect here. And what I can see is that we have now a one order difference when I compare to the protein 15. And you can say that we have here a cooperative effect of non-naive interactions. The last group of bar here is the experimental result, but if you compare to the last one, you can say that you have two orders difference here, but you need to remember that you are working with a simple model with a certain level simplification. Another result here is that you can avoid, I can say that you can avoid the internal friction by some mutation. Here we follow the same sequence of mutation that Joanne Clark made, just to change hydrophobic and charged amino acid to another groups of amino acids. And here we have the lowest folding time and the M5. M5, it's a combination of 5 mutation. And when you have this combination, you can get the almost the same root, almost the same folding rate to this domain. And here you can get the lowest ratio folding time to protein 16 in figure A, and in figure B, we have the same results to protein 17. If you remember in our last slide, I told you that you have such difference between our results, and a good way to compare the experimental and simulation results is in a logarithmic graph. And in figure C and D, we have not only the mutation that speed up the folding, but also the mutation that speed out the folding. And to the domain 16, we have a correlation coefficient 0.94 to this domain and 0.81 to the domain 17. And here you have a good correlation with our result with experimental 1. And you can say that the internal friction could be understood in terms of non-naive interaction. But the question now is, what is happening with the energy landscape of these proteins when I have this group of mutation? To analyze the energy landscape of this protein, I use the Alvin technique. Alvin technique is an energy landscape visualization method based on the distance of amino acids. And as a result of this technique, we have a 2D projection. Here we have example, the CI2 trajectory, and it dot here represents a structure, and the Euclidean distance between them is the structure difference. As can you see here, we have the reaction coordinate as function of fraction of neighboring contacts, and in red here we have the folded ensemble. Around then in blue we have the unfolded ensemble. Almost green and yellow, it's two possible transition states to protein CI2. But in our case, we have a combination of two trajectory, Y-type proteins 15 and 16. But if you take a look just to the protein 15 individually, we can see that our system goes from unfolded to folded ensemble through a low energy barrier, because here we have a high probability density in the transition state. But if you just look to the protein 16, you can see that our system goes from unfolded to folded ensemble through a high energy barrier, because here we have a low probability density in the transition state. And here you can see that what is the internal friction? What is the effects of internal friction in the protein 16? What is changing in its energy landscape? But what is happening when I have the set of mutation that speed up the folding? Here we have a combination of two trajectories too, but the protein 16 Y type and 16 with the 5 mutation. And here, if you take a look to the protein 16 individually with the 5 mutation, you can see that our system goes from unfolded ensemble to folded ensemble through a low energy barrier, because our system here appear with a high probability density. When I compare to wild type protein, you can see that our transition state here appeared with low energy barrier, that the low probability density. That's mean we have a high energy barrier. So as a final message, we need to remember that our first question, could you understand the internal friction in terms of non-linear interaction? And in this case, you can say that, yes, we can understand the internal friction in terms of non-linear interaction. And the kinetic effects can be explained in terms of cooperative effects of hydrophobic and electrostatic non-linear interactions. And this interaction modulate the energy landscape, as can you see in the last result, the energy barrier and the folding route of these proteins. I'd like to thank my supervisor, the postdoctoral research that helped me this last year, the Brazilian agencies, the region NESP at the São Paulo State University, and also the organizers again for the invitation. Thank you very much.
Thank you.
Okay, thank you, Fernando, for your talk.
No.
So now we have time for some questions.
Okay.
I welcome the audience members, and especially in the spirit of what we discussed yesterday, I would highly encourage students and postdocs to ask some questions first. Let's see.
Let me just ask a question.
Yes,
Can you
absolutely.
separate the electrostatic effects from the hydrophobic effects? Basically, you go from one coordinate to 2 coordinates, and that should help you. Is there a way of, have you done or not?
Could you ask again?
You are saying that these effects can much more hydrophobic interactions than electrostatic. Can you separate which one is dominant or how?
I don't have this answer right now, because I don't have this answer right now, Professor. But I could say that I think that the electrostructure is more dominant in this case, because my analysis showed me the result before, but I don't have the result right now to tell you.
Okay, that's what I thought, but thank you.
Okay.
Can I just make a comment to echo Arpita? So, and Simon Sponberg did a masterful job of this yesterday. The goal is for students to ask questions, and so we will sit in terrible silence until a student asks a question, was the Sponberg rule. And it takes effort to achieve that rule, but really we would like to hear from the students with questions. So
we have a few minutes.
Please.
Absolutely.
Please. And even if it's not your area, ask a question.
Okay, there is. Oh, there's a question from Clark. What is the typical length scale for these interactions?
Sorry, what is?
The typical length scale for these interactions? Like how far apart they need to be?
I think it's angstrom to nanometers. Yeah. Length.
Okay. And so, for example, the follow-up question is, are they local in nature? Or is there some extremely long-range effects, such as allosteric effects?
Sorry, but I didn't understand your question again.
So are they local? Are the interactions local? Or would there be some long range effects? Such
Oh
as
no, it's
electric
local. It's not long range effect.
So
It's
these
more
are local interactions. Okay.
Yeah. Yeah.
There's another question. Thank you. From Ria. What is the effect of solvent in these interactions?
The solvent.
Solvent, yeah.
Oh, but in this case, we have the solvent in your case.
Sorry, what could you repeat that?
But in this case, in my simulation, we don't have the explicit solvent.
I see you don't consider the solvent explicitly. Okay.
Yes, but we have the implicit solvent in the equation, but not explicit solvent.
Okay. Let me see if there's any other questions, or if anybody wants to speak up, raise their hand. We can allow faculty to ask questions now if they are so inclined. Let me see. Okay, so if there's no more questions, I'd like to thank Fernando for your very interesting talk. Thank you.
No, thank you very much.
I know we can't collectively applaud. Okay, so thanks so much. So our next speaker in the session will be Roger Zhu from Johns Hopkins University, and he'll talk about very fast Cas9 deactivation. So Roger, you can go ahead and share your screen when you're ready. Great.
Hi, everyone. Can everyone hear me?
Yes.
Okay, great. Hi, so my name is Roger Zhou, and I am a graduate student in TJ Haas Lab at Johns Hopkins University. And today I'll be giving a brief presentation about a new method we developed for very rapid deactivation of CRISPR using light. So just for a brief background, as I'm sure many of you know, CRISPR has been adapted for mammalian genome editing. And just a brief outline for the mechanism, you have a Cas9 protein along with a guide RNA whose sequence is complementary to the target DNA. And so there's in cells, there's complex formation followed by cleavage, which is subsequently followed by cellular DNA repair mechanisms to resolve this disruptive double-strand break, resulting in a possibly precise edit. There's also been interest in developing control of this mechanism, specifically spatial temporal control. And this can be achieved through a variety of mechanisms. One is through chemical control, and these are some example papers. Alternatively, one could have protein control to allow specifically here protein-based inhibition. And finally, there are methods to endow light control so that Cas9 can be, in the past, mainly activated using light. However, many of these methods have disadvantages. One big one is that many of these are multi-component systems. And so these are just 4 examples. There's no need to go into detail here, but as you can see, there are many different domains that have to interact to achieve the desired effect. And as a consequence, that can lead to very slow kinetics. And in addition, many methods exhibit incomplete activation or deactivation. So here, these are graphs from 2 papers where they show even with saturating doses of an inhibitor protein or a small molecule, there's still some pretty decent residual activity. And of course, that is undesirable. Now I will give a brief introduction to our method. In contrast to previous methods, we actually modify the RNA rather than the protein. And so in this guide RNA sequence here, we introduce a photocleavable group to this guide RNA. Now natively, so by just having that group there, the Cas9 enzyme is still able to make double-strand breaks and cut DNA. However, if we deliver light, that'll actually cleave this photocleavable group, making the guide RNA truncated. And it is well known that for Cas9, a truncated guide RNA is actually inactive. And so this allows a switch from an active Cas9 to an inactive Cas9. So in conclusion, what we have here ideally is a single component system. So the Cas9 in complex with guide RNA with a light-controlled off switch or kill switch in a sense. One of the first experiments we tested is in cells. So in this scheme, we deliver Cas9 with electroporation, so just shocking cells with electricity to proteins to go inside. And then we could either shine light at many different time points. So what I'm showing here is either shining light right before delivery. Ideally, that would completely deactivate all Cas9. Or we can shine light at an earlier time point. And the expectation there is the activity will be greatly reduced. And then for all these samples, we will always harvest at 3 days later, all by sequencing, to check for the editing. So what we see here is, I will direct you to this untreated sample. So what untreated means is these are just standard untreated cells. So naturally, there's no editing to be observed. Inactive corresponds to shining light before actual delivery. And so the expectation here is during delivery, all the Cas9 should be inactivated. And what we do see is there is almost no basal activity. If we shine light at a pretty early time point, you see there's a significant suppression of editing. So the y-axis is editing, measured as a percentage of insertions or deletions from sequencing. And however, if we do not shine any light, then the editing efficiencies are actually comparable to a wild type guide RNA. So a guide RNA with no modifications whatsoever. And so with the system, we have first significant deactivation by light that is complete. In addition to that, we have very high editing efficiency when we do not use the deactivating light. Because this system uses light, we thought we could test whether we can allow spatial control over deactivation. So in our experimental scheme here, we first deliver Cas9 to cells, and then we shine light in a very specific pattern, followed by transfection, so separate delivery of a reporter that normally results in GFP, so green fluorescent protein expression. However, this reporter has a Cas9 targeting site. So cells that have active Cas9 will destroy the reporter, and so will not express GFP. So this is a cool example of our results here. The top row shows the masks we used. So the white here corresponds to areas where the light can shine through. And so this corresponds to areas where there is GFP, because the cells that are aligned here have not have had their Cas9 deactivated. And so the reporter system can act as usual. The next question we wanted to address using our system is to better understand DNA damage and its repair. One question that has been difficult to answer with other methods is, after there's a double-strand break that's induced by either naturally or by an enzyme like Cas9, how long does the repair last? And more specifically, how long do repair proteins last? Because it is well known that many repair proteins get recruited to sites of double-strand breaks to facilitate repair. There are two previous methods for accomplishing this. The first one is with laser microradiation. So you can see you just zap a cell with a very powerful laser. The advantages here is you have a very precise start, and there's no additional damage. However, the damage can be very complex, and of course, there's no sequence specificity. Oh, and in addition, you can only do one cell at a time generally. In contrast, we can use endonucleases such as Cas9 to make sequence-specific cuts. And here you can cut multiple cells at once, of course. However, there's a crucial issue here in that, so this can be visualized in this video where the red corresponds to a DNA repair protein. And what you can see is there are multiple cycles. If you just track this one here, see there's multiple cycles of recruitment followed by dissolution. What that corresponds to are actually multiple cycles of DNA damage and repair. Now that is because once the DNA is repaired, generally faithfully, there's still active Cas9 which can make further cuts. However, that deconvolutes the kinetic process that we're trying to end, and so we are unable to answer the question, how long do repair proteins persist for one round of repair? So the way we use our system, which we're calling pcRNA, for photocleavable RNA, is we deliver Cas9, and then we let it sit inside cells in its active state for a period of time. And then after we deactivate at time 0, we can harvest and cross-link cells so that we get cells at different fixed time points. And then we do chromatin immunoprecipitation for specific DNA damage proteins, followed by sequencing. So this whole pipeline is called ChIP-seq. And the goal of this pipeline is to map protein binding sites on DNA. More specifically, in our case, DNA damage response proteins. So this is an example of the results we get from here. We're looking at MRE11, which is one such protein, which localizes very right adjacent to a cut site, which is marked by this vertical line. And these intensities correspond to enrichment of MRE11 surrounding the cut site. And this horizontally is a 5 kb region centered at the cut site for the actin gene. 0 minutes corresponds to when we shine the deactivating light. And so there's already some enrichment at 0 minutes. And then interestingly, after deactivation, there's still some increase followed by an exponential decrease. And by 2 hours, the majority of the enrichment's already gone. We can look at many different proteins. This is a different example of 53BP1. So this protein spreads actually megabases. Here we have 6 megabase range. And in contrast to MRE11, this actually exhibits a linear decrease over time, starting from the deactivating light. This shows the quantification for both MRE11 and 53BP1. So in summary, what we have is a very fast and precise and complete CRISPR deactivation system that uses light. It is a single component system, which we can call as a built-in kill switch or built-in off switch. And we're using this system to study DNA damage and repair. So I'd like to thank TJ for his great mentorship, as well as Yang and Binwoo, who are a postdoc and another PI with collaborating, as well as all members of my laboratory. Thank you.
Great, thank you, Roger. Very nice talk. So we have, that was just in time, so we have time for questions. And as Dan urged again, hopefully we'll get questions from students. So we already have a question here in the chat from Kevin. What light wavelength did you use? Are your results wavelength specific, or a broad spectrum of light will lead to the same results?
Yeah, great question. We used 365 nanometers. So that's an optimal wavelength for cleaving the photocleavable group. However, the wavelength or the intensity is actually quite low. So for these experiments, we just use a black light that you can buy from Amazon.
Okay. But just to reiterate the question, will are there other wavelengths that would also work, or is this?
I think the optimal wavelength was around 365.
Okay.
We tested in the 400s range, and that does not work as well.
Okay.
Is it okay if I just ask my question?
Yeah.
So relating to that similar question, but so around that wavelength, are there any other concerns of anything you might disrupt in the cell at 365 nanometers?
Yeah, so depending on the power, we've observed some slight growth arrest that's pretty maybe within a day, but then things go back to normal, and we do not see additional cell death. But that is certainly a concern because this wavelength, of course, could be toxic depending on the dose.
Any other questions, either by chat or speak up or raise your hand?
Are you ready for faculty questions, Arpita?
Any other students debating on a question? Okay, so I'll let faculty ask questions, but definitely students can come back with more questions. Go ahead.
I had just a general question. Could the same methodology of basically your photocleavable RNA be used for other cases of RNA regulation? For example, a microRNA that suppresses translation, again, has a sort of matching capability, which is sort of similar to the way CRISPR guide RNA works.
Mm-hmm.
So have you thought about using the same technology for many other RNA regulation systems?
Yeah, so this, just the concept of using this photocleavable group could be extended to, certainly, as you said, microRNAs or other methods of RNA regulation. And in addition to that, we could also use it for other CRISPR proteins as well, because all of them use RNA guided, or are RNA guided. But yeah, so that is certainly an area that we currently have not started working on, but we think there's great potential to use this photocleavable system for those purposes.
Great.
Thanks.
Thanks.
Yeah, thank you.
Other questions? You probably have half a minute, so maybe I'll ask a quick question and
Sure.
then. So I was curious about the photocleavage efficiency because there was some residual activity still after photocleaving. Is that?
So I don't have the data here on the slide, but we see that with 30 seconds of light, the photocleavage is actually complete.
Okay.
And that is the duration that we actually use in cells. And yeah, so I guess even though statistically it's not significant, there could be some very low basal cleavage, but we think that could just be because, not necessarily because of incomplete inactivation, but there's still, even though it's inactive Cas9 in the system, it's possible that a truncated guide RNA could potentially lead to some very low levels of cleavage. That is unclear.
Okay, great.
Yeah.
Thank you.
Great, thank you.
Okay.
Thank you, everyone.
If there's no other questions, I'd like to thank Roger again for a really interesting talk. Thanks so much for sharing your results with us. And I will now give the moderator role to Ilya, I think, who is scheduled to moderate the next two talks. And yeah,
Thank you.
we'll see the rest of the speakers on Friday. Yeah.
Thank you, Arpita, for being our moderator in the AM under trying circumstances. Appreciate it.
You're very welcome. And I'm glad that we were able to give the students a chance to speak, and we also have a good audience. Yeah.
I'd like the next speaker to start connecting at this point. And my name is Ilya Nemenman. I'm a faculty here in physics at in the Department of Physics and Biology at Emory University here in Atlanta. And we're part of the same PoLS node as Georgia Tech here in Atlanta. And so the next speaker of the session is Joanne Chen. from Bill Bialik's group at Princeton. She's going to talk about searching for emergent long time scales without the fine tuning.
Okay, thanks, Elia. Can everyone see my screen and hear me?
Yes, we can hear you, yes.
Okay, that's great. Hi, everyone. My name is Joanne Chen. I'm a graduate student at Princeton University working on biophysics questions. And thanks for the organizer for bringing together this iPost conference online. And I'm very excited today to talk about my work with my advisor, Bill Bialik, on emergent long time scales in systems of neural networks. Okay, so first, what do I mean by long time scales? As we experience every day, most of the human and animal behavior occurs at time scales that are much longer than the intrinsic time scale of individual neurons. So for a single neuron, if you're considering the action potential and the refractory period, it takes about 10 milliseconds, so on the order of milliseconds. On the other hand, for example, even for you just to understand my sentence, you need to hold the information of the beginning of the sentence in your mind for example, order of 10 seconds. So this is already an increase of 10,000-fold in time scale, which is quite amazing, but not something that we really think deeply about every day. So in some systems, it's found that these memory traces are encoded in firing patterns that is sustainable over a long period of time. So here is just an example in goldfish oculomotor systems, where these persistent neural activities last over 1 second, and those firing rate encodes head position of the fish. So how can we explain this long time scales? One of the first places people have searched for an answer is just the mere fact that neurons are connected. And the simplest model to explain this idea of emergent long time scale in neural networks is just a linear dynamic model, where we can consider the coarse grain variable being x, the firing rate of neuron I. Each of them have an intrinsic time scale of decaying as 1. They're also connected to other neurons with this matrix M. And with this linear dynamical system, you can solve for the dynamics just using eigenvalues lambda and eigenvectors R and L of this matrix M. So the time scale of the system is really just 1 over the difference between 1 and the lambda, the eigenvalues. To get the system to have a very long time scale or slow mode, what we need is the largest eigenvalue lambda max to be close to 1. But here is also where complication comes out, because we also want the system to be stable, and then that requires all eigenvalues to be less than 1. Otherwise, it's going to blow up over a long time period. So in order for the largest mode to be both slow as well as the system to be stable, it requires fine-tuning of the matrix elements M. And in particular, if you want a system to have 10,000-fold of increase in your time scale, this element of connection matrix need to be tuned with an accuracy within 0.01%. And another limitation of this approach is that it doesn't differentiate the situation where there's a single slow mode or a continuous spectrum of slow modes. Both may be important for the long time scales we observe in actual behaviors. So today in my talk and your work, we asked the question, can neural systems generate long time scales without fine tuning? And specifically, our goal, writing down the equations, is really to find a distribution of these matrix P of M such that this longest time scale, T max, grows as system size M, okay? All right, so what's our approach? So the simplest distribution we can choose for M is really just for M to be symmetric. We notice that it's not accurate for real neural networks, but it's just simpler to study. And we also choose the matrix element from a Gaussian distribution here with a variance of C² over N. So C is really the interaction strengths that you expect on average, okay, how does one neuron connect to the other? And because this distribution is invariant under orthogonal transformation, you can actually solve for the eigenvalue distribution and just a Gaussian plus some log repulsion among the eigenvalues. And using mean field methods, you can solve for the spectrum density. And this is the well-known semicircle law by Eugene Wigner in the 50s. As we can see here, the support of the semicircle or the spectral density is a function of the interaction strength C. And this implies that there are 3 different regimes depends on the strength of the interaction. So if C is small enough, then you have basically the semicircle away from lambda equals 1, which is where our systems becomes unstable. If your eigenvalues is larger than that, then there exists a finite gap. So you don't have long time scale. Okay, but if your C is too large, then you are in the unstable regime and your system will blow up. Eventually, this is saying that you will only get slow modes at the critical interaction strength C equals 1 over square root of 2, where you have the spectrum density tangential to lambda equals 1. So notice that this is a fine-tuning, but this is also a different fine-tuning that I mentioned earlier, because earlier we are really fine-tuning each matrix element. And here, because we are looking at the distribution, we're really just scaling the entire spectrum density. Okay, so can we now do better by maybe changing the distribution of interaction matrix by adding some global constraint to the Gaussian interaction? So the first thing that we tried is really to look at this unstable case. And we can imagine that maybe a biological system through evolution or some biophysical process can really constrain itself to be always stable. And mathematically, it is just to put and constrain such that all the eigenvalues are required to be less than 1. And the resulting spectrum is recently studied also in random matrix theory by Dean and Majumdar. And what they found is quite interesting, which is basically the spectrum density diverge near the wall. And correspondingly, the longest time scale T max grows as n squared as system size. So here we can indeed get long time scales without fine tuning if our interaction strength is large enough, and there is a definite way of knowing if the system is stable or not. Okay, but here maybe you see the problem. And the problem is just this hard stability threshold is not biologically possible. So for a system to know for sure if it's stable or not, you need to wait an infinite long time, okay? So what can a neural network do such that it can tune the stability in a biological plausible way? Okay, one idea is just to monitor the neural activity using the time average of the norm. If the system is going to blow up, the norm is going to increase, right? So if we just constrain the norm, then we have a handle on the stability of the system. And in addition, we can recall that neural activities are intrinsically noisy. And in case of a white noise, the neuron activity has an average norm that depending on just the eigenvalues in the steady state. So what we want to do is have a distribution that constrains this norm. And there are many distributions that does this. And among those, we choose the one with the least amount of additional assumption. And so that's just to maximize the entropy of this distribution. And we still want to draw the matrix element from a Gaussian prior. So this modified distribution of eigenvalue is you have this Gaussian prior multiplied by the exponential of something times the constraint that we're putting, okay? And then here, Xc is just a Lagrange multiplier such that we fix the norm to a constant. Well, how does the spectrum look like now? We modify this hard wall to a soft wall with a 1 over x decay from this, this constraint on the norm. After solving it with mean field method, what we see is that, okay, first the spectrum no longer diverge, it turns down. And interestingly, there exists a finite gap between the rightmost edge of the spectrum and lambda equals 1. So what does that mean? Well, it means that now the longest time scale, which is just inverse of this gap, is fixed. It's upper limited, and no matter how large your system grows, you will not exceed this long time scale. And we can confirm this by comparing this mean field result with Monte Carlo simulations, which is finite system size results. And what we found on the left is indeed, if your system is increasing, you do have an increasing Tmax, but it's upper limited by this mean field result. But on the other hand, we can also think about it is if our Lagrange multiplier is small enough, then if your system size isn't too large, then you still have this n² increase in your Tmax. So you kind of recover this increase in the growth of longest time if your system isn't too large. Okay, but all these are plotting against this Lagrange multiplier Xc, which is not a biological or physical quantity. And the physical quantity is really the norm. And we know that the norm will actually increase if your Xc is small. So here what we're showing is exactly the plotting Tmax against the expectation value of the norm per neuron. And what you see is basically Tmax keeps growing as the expectation value of the norm grow. And practically what this is saying is that if we want our system to have a long time scale, we need the system to be able to tolerate a large norm, or equivalently just have a very large dynamic range. And in fact, this dynamic range doesn't need to be too large. So for example, if we look here, if we allow it to be just 10 times what we expect from an independent system, you can already get a 10,000, and 1,000 folded increase in your time scale. And just to complete the picture, I'm also showing you that at this critical interaction strength C equals 1/√2, you can achieve an infinite long time scales with a bounded dynamic range of just 2. So to conclude, what we show is if you have a hard stability constraint in this linear dynamic model, you can have divergently many slow modes, but it's not biologically plausible. And if we now tune it to be using a soft constraint on stability and with constraint on the norm of neuroactivity, we can have the longest time scale set by the allowed dynamic range. And currently we're working on figuring out the synapse dynamics that leads to the norm constraint distribution. Okay, so thank you very much for listening, and I'll take questions.
Thank you, Chairman. Thank you. Everybody, the floor is open for questions. Just unmute yourself and ask them. And we'll start with graduate students, postdocs as always.
So you showed these results from Monte Carlo simulations. Can you do some finite size scaling there, or somehow otherwise collapse the data that you showed?
So I think when I collapse the data, it depends on C. This interaction strength. I mean, I tried and it doesn't seem like it collapsed for all. I mean, I think there is more parameter values we need to tune.
So you would need to include the Xi in the scaling somehow.
Yeah, I think so.
I see. Okay,
yeah.
Thank you.
Do we have any other questions from students? Okay, I'm going to open the floor for faculty.
I don't know if you want to, but can you say a little bit more about your last part? I mean, how does synapses, which I think of as being, local, how is that going to affect something which looks like a global constraint on activity?
Yeah, So when you're looking at synapse dynamics, sometimes we usually think about just hidden learning, which is a local operation, but there also exist global scalings of synapse dynamics, such that, for example, you globally rescale the synapse strengths overall, or you modify this hidden learning with some threshold that is based on, for example, the neural activity. Okay, so I can talk a little bit about the current work, which is basically when we're looking at dynamics, okay? And eventually what we found is a combination of hidden learning with a threshold that is based on the neural activity and the overall neural activity globally. And how can a system achieve that? So in some system, you can do it through neuromodulators, which affect like a certain region through diffusion.
Okay, thank you.
Okay, we have another question from Alexander Smirnov.
Hi, I'm from University of Maryland, and we're working on brain initiative. That's very interesting, this study. My question is just, could you clarify for me the model for this neural network? Is it all excitatory neurons? Or, I mean, I imagine there are also inhibitory neurons in real brains, let's say.
Yeah. So here, what we're using is this connection matrix, just choosing each element randomly. So it can be positive and negative, but it is not precisely this excitatory neuron or inhibitory neuron, because those you would expect, let's say maybe the entire row or a column of this matrix having the same sign. We do not have this, this requirement on our model. I think it's very interesting to add this constraint and then having more realistic models and then see how it changed the spectrum.
Thank you.
Thank you very much. And I think we are right about out of time. So thank you very much.
Thank you.
And the next speaker, would you please share your screen now? And I think this is Basilio Chiazza-Quaman.
Yes, yes, I am sharing my screen right now.
Pronounce the name correctly.
Yes, my name is Basilio. Thank you very much. Can you see my screens?
Yes, we can see your screen, and so I'm giving control to you. Basilio is from Johns Hopkins. Thank you.
Okay, thank you very much for the invitation. Today, I'm gonna be talking about a computational project that I has been working in very close with Professor Elijah Roberts and Krashan Blakov. Here we develop a reaction diffusion model to study phase separation. For people who is not familiar with phase separation is the conversion of a single phase system into a multi-phase system. For example, here you could see protein concentration versus time, and you could see how the concentration of the protein increase and then decrease. But there is in this system a critical concentration where when the system is above the critical concentration, the system switch from a diffuse state to a condensate state. And as long as the total concentration of the system is above this critical concentration, the droplets formed in the condensate state are stable. In this next plot, you could see that some external factors like the post-translation modifications or change in the temperature or ionic strength can modify this critical concentration, can lower the critical concentration so that favoring the phase separation. Why are we interested in this problem of phase separation? There is a lot of biological systems that show phase separation properties or look-alike properties. For example, in this work, the authors show that P granules have liquid-like properties. Here, for example, you could see the nucleus, and in red, you see the P granules, and on their shear, you could see that the P granules drop from the nucleus like liquids, droplets. Also, other studies show that nucleoli can coalesce and form bigger droplets. And in other words, also people has induced the nucleation and growth of nuclear bodies in the nucleus of Drosophila oocytes. The method that we're going to use is the reaction diffusion master equation. The reason we are using this method is because we want to get information about the number of particles, but also the distribution of the particles in the space. How we're going to implement the reaction diffusion master equation using the software or the toolkit Lattice Micro developed by Professor Elijah Robert in 2013. The model consists of two species, X1 and X2. And the dynamic of the system is described by these two reactions, where X1 and X1 interact to form X2 in the first reaction, and then X2 recruit more X1 to form more X2. A little bit more of the detail of the model. We're going to do the simulation in this volume where we have 1 micrometer by 100 by 100 micrometers. And here we're going to simulate the reactions. These are some of the typical values of the parameters that I used during the simulation. So the first question that we wanted to answer is, can this model show phase separation? So what I'm gonna show you here are the results from one simulation where in the left, you're gonna see the species 1, in the right, the species 2, X2. And the important here is that both are the same simulation, just that here I'm gonna focus in the species X1 and here species X2. The dimension is 100 by 100, as I mentioned, and this axis shows the number of particles. When you see black, means that there is 0 particles. When you see white, means that there is a maximum of 3 particles. As you could see in the beginning, we started simulation with the whole system just formed by X1 particles and no X2 particles. And what we found is that X1 doesn't phase separate, but you could see that X2 phase separates. So from here, we can conclude that we have developed a new model that shows phase separation through clustering formation. Obviously, the dynamic that I show you just correspond to one specific point in the space of possible behaviors. Here I'm gonna show you 3 other parameters or the behavior of the system at 3 different parameters. For example, here you could see the cluster that are more dynamics and noisy. In the second one, you could see that the clusters are better defined, although they are still very dynamics. And you can find regions where basically the clusters are very dense and stable, and they don't diffuse as fast as in the previous simulations. Obviously, we can continue changing the parameters till we form a phase diagram. In this phase diagram, I'm gonna show you K1 versus K3, and every point in this phase diagram corresponds to 20 independent simulations. The average of 20 independent simulations. K1 corresponds to the nucleation rate, K3 corresponds to the cooperativity strength. And the phi parameter is a parameter that gives you information about the degree of clustering in this space. As you could see, there are different regions in the space. For example, let's focus right now in B. Again, this dimension is 100 by 100. Here is the phi parameter. And you could see the X2 form, but they don't cluster. However, if you go to the region in C, you could see that in this region, you're gonna find several clusters, small clusters. In general, they are very stable here. Then in the region D, you could see that the clusters are bigger, but also they are amorphous, more amorphous than in the previous one, and also they are more dynamic. And finally, there are other regions where all the X2 particles collapse in a huge clusters. So from this result, we conclude that the clusters in this phase separated model show different dynamics and size depending on the nucleation rate and the cooperativity strength. So the next question that we were wondering is, does phase separation in our model occur in or out of equilibrium, or both? So to answer this question, we did a detailed balance analysis. The detailed balance analysis states that in equilibrium, the flux of particles from left to right should be equal in each reaction, which is stated in this expression. J corresponds to the flux, plus correspond to the direction of the flux of particles from left to right, minus from right to left, and I correspond to the reaction, the first reaction and the second reaction. So when we apply this relation to our reactions, we end up with this, with this relation where K2 times K3 divided by K1 times K4 is equal to 1. And what this means is that phase separation in our system occurs at equilibrium when 2 is satisfied. Otherwise, the system is out of equilibrium. So what we did is, based on this expression here, we keep constant K2 and K3, and this we start to change the, well, sorry, we keep constant K1 and K4, and we change K2 and K3. And we get this plot. Here you could see a region in a white triangle. This region corresponds to the region where you see clustering. And here there is bistability. In other words, the system can switch from a diffuse state to a condensate state. The region out of the triangle is all the time in a diffuse state. To test if the system is in equilibrium or not, we calculate the difference in the chemical potential of converting two X1 particles in one X2 particles. And if the system is in equilibrium, this difference should be 0. In other words, should be in white color, as you could see here along this line. And if you compare with a region in where you see bistability or where you see clustering, you could see that the region of clustering never crossed the region of equilibrium. So, and also it is in the region where you see a difference of chemical potential of 20 or 15. So from here we can conclude that phase separations occur out of equilibrium. The next question was, does nucleation promote phase separation? Previous works, for example, in these two works, the authors, for example, in the first paper, they isolate a 12-base pair DNA sequence. And with this sequence, what they did is basically localize this sequence in a specific position in the genome. And they show that this sequence promotes clustering. In the second paper, the author did something very similar, but instead of this DNA sequence, they use a protein domain. And also this protein domain promote the clustering. So if we see the change of diffuse state to the condensate state, like a 2-state model, we can suggest that maybe the nucleation is favoring the transition by decreasing the amount of work that you need to transition from diffuse state to the condensate state. So how we implemented the nucleation? As I explained before, our system consists of these two reactions, but we added a third reaction to the system. This third reaction is basically the same as the previous one, the second one here. But the only difference is that we're gonna introduce a particle X2 star. This particle X2 star is the same as X2, but the difference is that the diffusion coefficient of these particles is 0. In this way, we can localize the X particle in a specific location, like in the previous experiment that I told you about in the two papers. The K3 and K4 is exactly the same as the previous reaction. And in more detail, what we did is, here is the volume where we did a simulation. And in this location here, we introduced two X2 stars with a diffusion coefficient of 0. And then we add an amount of X1 and allow the system to equilibrate. So what we found is that, as I showed before, here we have the nucleation rate versus the cooperativity strength phase diagram. When we introduce fixed nucleation sites, you could see that a region that previously didn't show clustering or phase separation, now is showing phase separation. So from here we can conclude that fixed nucleation sites promote phase separation in our system. So to conclude, we have developed a new model that shows phase separation. Clusters show different dynamics and size depending on the parameters, in this case, K1 and K3 that correspond to the nucleation rate and the cooperativity strength. Also, our system showed that clustering occurs out of equilibrium. And finally, fixed nucleation sites promote clustering. With this, I want to finalize giving thank you to Professor T.J. Ha for his mentoring and patience with me, and also mostly to Professor Elijah Robert and Kristen Blago, who trained me, mentoring me, and teach me a lot about dynamical system and computational simulation, and also to Professor Sua Mion, with who we have very exciting and interesting discussion, and she also gave us advices about the phase separation system, and finally to all the HALA members. And I will be happy to answer questions.
Thank you very much. Thank you for this great talk. And as always, we're opening the floor for questions from students.
Yeah, so I have many questions, but I'll limit it myself.
Yeah, could you please introduce yourself before you talk so that we know who's talking?
I'm sorry. My name is Zane Thornburg. I'm a student with Zan Luthi Schulten. So Elijah's grad school PhD advisor. But anyway, so your reaction network, why is that this particular set of reactions that you chose?
Oh, yeah. First of all, we're gonna publish this paper where we really found a set of reactions that show this behavior. I mean, the reaction that I show here is just a very particular example of a set of reactions that we identify. So this is just a minimal case that we are showing that show this behavior, but when we do a generalization of this model, we can still see phase separation. So in other words, we identify a set of reactions, and this is just a simple example.
Thank you. And there's a bunch of questions I see on the chat. So I see Michael Martini is asking, was there a diffusion rate in the first model? Does this help set the length scale of the patterns, or how is the scale set?
Okay, diffusion, yeah. For example, in this simulation, in most of the simulation, we use this diffusion coefficient that I'm gonna show you here. Okay, for example, for the particle x1, the diffusion coefficient is this one, and for the particle x2, the diffusion coefficient is this one. Now, obviously there is a big difference. However, we have done this simulation, a different diffusion coefficient for x2, and even though when the diffusion coefficient for x2 is very close to x1, we still see clustering or phase separation. However, as the person who asked the question intuitive, yes, the diffusion coefficient, the difference in the diffusion coefficient gonna influence the dynamics and some of the properties of the system. Yeah. Well, the reason x2 we decrease the diffusion coefficient is also because several works have shown that when the particles form the condensate state, in the condensate state, the diffusion coefficient is slower than when it is in the diffuse state.
Okay, thank you. So the next question comes from Fritjof Browns. Have you tried a mean field description of the system? Does the clustering depend on stochastic effects?
No, we didn't try any mean field description, and the clustering depends on the stochastic effect. Something that we found is obviously that the switching gonna depend on the size. When the system is very small, it's more easy to find a switching from a diffuse state to a condensate state. However, in the publication that we're gonna release soon, we did a size effect, how the system depends on the size effect. So we start with very small simulation, and then we increase the size till we did a very high system. And even in the very high system, big system, we still see clustering and also the phase transition.
Okay, well, thank you very much. I think we are right on time right now, 15 minutes from the start of your talk. So thank you very much for your talk. And you can only hear me clapping, but I'm sure everybody else is joining as well. And I think that this ends the session, and I'm going to cede the microphone to Margie and Daniel, who are going to tell us what's going to happen next.
All right, okay. Well, go, Dan, you go. Okay.
Sorry, sorry, I was getting distracted by something else. What am I supposed to go?
What's the plan now? So I know the.
Yeah, I think that if we want to continue a discussion, we could do so now. like I said in the email, I'm not prepared to lead a discussion, but if folks want to talk, discuss, reflect, feel free. And if not, you're welcome to leave, and we'll see you tomorrow.
On my behalf, this is Margaret Chung. Hi, can you hear me?
Yes.
I would like to share some sentiment here as an outreach director at the Center for Theoretical Biological Physics. We work with many students from underrepresented group, from African American community, from people of color. And I can tell you that the pain is real. And I myself is an immigrant. So therefore, I did not really experience these oppressing experiences when I grew up. But however, I live in Houston. Houston is deep South. This is where Jim Crow laws had happened until late 1960s. So the legacy exists. And University of Houston, where I worked, is in the 3rd Ward. And this is where Blacks live. So how did that work is that during that time, there is ways to segregate community. If you come to Houston, you see there is a highway from north to south that justly splits Houston in half. And in the east of Highway 288 are Black and poor. And the west of 288 is mostly rich and mostly white. And how would that impact education is because we know schools are supported by property taxes. And because they're poor, their property taxes are low, and they underfund their schools and resources and healthcare. So these legacy propagate or pass it down to the next generation. So we have, UH is on the 3rd Ward, and across the street is the Yates High School, is where George Floyd, studied. He's a football star there. So it's really hit home in our sense is because 3rd Ward has been our partner at UH to fight systematic racism. There is another university that is right across the street, that is Texas Southern University, historical Black university. And we worked together. We wanted to bring a lot of the resources that most students here think of for granted, but they have never had a chance to experience. So I thank the NSF for all the outreach activity that sponsors us, help us, train us, and teach us how to communicate science and how to understand such unfair, such pain that systematically oppress the Black people. And over here in the audience, we have some FIS students. FIS students, would you like to share some of your stories or some of your past experiences? I don't know if some of you're here.
Margaret, why don't you explain what FIS is for the general community?
Okay, so I'm sorry. So FIS represents Frontiers in Sciences. And this is the outreach program that's affiliated with the Center for Theoretical Biological Physics. So what we do is that we recruit students from UH and nearby minority serving institute. UH has more than 45,000 students, is a Hispanic serving institute. More than 25% of the students are full-time from Hispanics. And also we have 10% African-American students here. And most of our students are disproportionately first in the family to attend college. So when they come to college, they don't really know what major to be because they're first in their family to enter college. They may think about going engineering or going to some majors that will see immediate financial outcome. And not many would consider science as a viable major. And in addition, most of them has to work because they are financially stressed. Most students at the UH, they have to work more than 20 hours per week in order to work on minimum wages to make ends meet. Whereas students from affluent community would afford to do research for free. The students at UH, they may not have this luxury. They may have to work as a clerk or work at the retails instead of working in the lab to improve their chances in science. So this is where the FIS comes in. We reach out to students from Houston Community College that have more than 70,000 students who are, some of them will transfer to UH. And some students from UH that would be interested in majoring in science but didn't know how to. We group them, create a cohort, and train them. We have mentors from the CTBP, mostly are senior graduate students and postdoc aspiring faculty. They join me to reach out, to talk to students, to give them, to basically provide mentorship. And over the summer, they will conduct research at the CTPP at Rice University and give one-on-one mentoring, understand what is the gap in their background, and then help them to bring them up, to help them leverage the field. And as a result, our program has been amazingly successful in bringing students of color to go to prestigious graduate schools countrywide. So I encourage all the faculty who are interested in outreach, reaching out to minority. I would love to share a lot of our successful experiences. And also I thank Jose and Herbie and Peter and Kristan for the leadership in making this happen. Thank you.
Thank you, Margaret.
Thank you, Margaret. This is Arpita. That was really great to hear about your efforts. And I think we can spend some time thinking about and learning about how each of us can engage in this effort. So it would be great to hear about your experiences and also learn from it.
I think one of the very important is the GPA issue, because the students who are really work very hard, but unfortunately they're financially stressed. So their GPA usually are not stellar. Their SAT are usually not stellar. If there's a graduate program that use GPA or SAT, GRE scores as a first round of cutoff, they may not appear on the top. So I would say that, you know, if we wanted to, we actually have a hypothesis going on at the center, is trying to correlate what is the factor that defines success in FIS students and see if GPA is a good indicator. So far, GPA has really correlated very little with to predict student success in graduate school. And I also saw Basilio. Basilio has worked with me before, and he's amazing. And Basilio, do you want to share your experience when you're working at UH?
Hi, Margaret. Thank you very much. Sure. To be honest, I agree with Margaret in all what she said. For example, in my experience, I had the amazing opportunity to work with Margaret, even though I had huge difficulties with language. Also, for example, at least me as an international student, for example, when we want to apply to a grad school, we have several barriers. One is the language, that of course everyone have, but also when you take an exam and your the English is not your first language, first you have to understand what you are reading, and then you can start working on the problem. And so I think that is really harder. Mostly, for example, in my case, I started learning English when I was 22 years old because my family was really poor and I have to study the school basically at night. And I have to leave the school when I was a child, so a kid, and I studied in the school when I was basically 17 years old, and I finished my high school when I was 21. I started my undergrad when I was 24, and I started a PhD when I was almost 29. So yeah, I agree with her that for us, sometimes could be really, really hard. Someone like, for example, Margaret Chan, Martin Rubel, also Professor Mario Ansel, who in the beginning, even though my English was really bad and very difficult to understand and work with, they make a huge effort to help me and to improve my skills. And well, that's the reason I am very grateful with every one of them, and I am completely agree with what Margaret said. Well, that's all what I have to say, I think.
Thank you. And I also see there's a FIS mentor. Vinny, you're here. Vinicius?
Hi, Margaret, how are you?
Your past experience in participating in FIS. You gave a fantastic talk at the UH a couple of years ago. Do you want to share with the community, how do you reach out to minority students?
So yes, sure. I have the opportunity to be a mentor of 5 students, I think, and was great. So a lot of diversity during the summer. So it's a trade-off. So it's good for the mentor. So I learned a lot about how to teach, kind of start to have those PI problems about how many students can I keep. And for the students, it's also good because we have a study group. They learn a lot about how to write, how to give a presentation, how to perform research. In general, that they are very hard work, and having this time during the summer only to do research, it's kind of shed the light if they are to pursue a PhD or go to work. So this is a very nice problem in general.
Thank you very much. This is how we share our experience with the community. If you need more questions, we'd be happy to address them.
There's some questions in the chat. As a new graduate student, I'm curious what we can do going forward to expand inclusion and make a career in science seem more approachable. Would anybody be interested in answering?
Hi, may I make a comment?
Yes.
No, well, I'm Victor, I'm from Brazil, and I would like to congratulate you for this effort. Margaret, in particular, your words are very, very applicable to Brazil as well. So, wow, the conditions in Brazil are far worse than those that you have over there. And this initiative that you have in with PoLS is fantastic, because our students in Brazil are reached as well. So you saw today presentation by Fernando. Tomorrow there's going to be another student. So I thank you so much for this opportunity to reach out other students abroad as well. So it's so nice, and I thank you so much.
So
Peter, I'll say something really fast. This is Simon Swanberg from Georgia Tech. I think these kinds of questions are exactly the kinds of things that are really good to ask, and we have opportunity as a student research network to try and do some things on this. I think when we're trying to address these considerations, we should remember that this is a this is a network for the students and ultimately run with the students. And so I think that there's a lot to be said on this. many of us are, I don't consider myself certainly an expert on how to do this, although we do have some activities that we're trying to do with mixed success at times. But I think it would be, I'd love to hear from the students and the postdocs as well about what they think we can do as a network and what we as a community can go for. That's not to sort of abdicate responsibility. I think that's a very important thing, but I think for issues like this, it's very important, especially in our group, to sort of hear from everybody and think about how we can all move forward. And that may not happen immediately today, right? This is, as Dan and various folks said at the beginning, this is sort of, I think a lot of things have been changing, and this is a very dynamic situation that's going on right now. So it's nice to give space to the stuff, but perhaps we can all think a little bit about how we might make this serve as better for everyone. My one thought in this is that, and I appreciated what Margaret was saying, is, you know, reaching out and providing new opportunities and actually actively recruiting people into the system seems to be very necessary. If we set up sort of passive systems that just sort of open the door if people reach out, that seems to not have nearly as much effect as if we actively try to reach out and do promotion and get the news out for these activities. I know we provide paid research positions, but without advertising, it tends to still be people who sort of know to go after those opportunities that end up seizing them. So sometimes raising awareness of what opportunities are out there is very important.
Yeah, I just want to add that absolutely. I think we can think about this as a wake-up call because many times, you know, people get busy with what they're doing, but we can use this time to really think about what actionable things we can do. And maybe if each person can think about one specific action they can do, and as you said, reaching out actively to schools, both at the undergraduate level, but also even in terms of K-12, for trying to actively recruit underrepresented minorities and giving them opportunities in our education systems, in our research systems. And maybe we can think about one or two actionable things that each person can do. I think that would be good. And then we can have a discussion or some sort of a site where we can share resources to find out what are these institutions in each neighborhood that we all are working in locally. And it'll be great to hear from graduate students and postdocs too, how they feel comfortable in terms of how they may be able to promote these activities. So I see some more interesting comments that I think everybody can read.
Can I just make a fast comment, Arpita?
Yes.
As people saw from the email sent by Margie yesterday, the entire Feast of Living system and our programs have been very involved on that. I'm very thankful for Margaret for her presentation, talking about what we do at the center. That's her leadership. That's something that's achieved. Thanks for her to be in the leadership and for the students and postdocs that participate on the event. But I want to point out that on the letter that basically the PoLS network believe that we should organize an event on that in a way that we are more planned for it. This came so fast. That's such a sensitive and very difficult issue and very important issue. So read the email and remember that on the end of the meeting on Friday, we are going to try to start to organize this event. I don't know exactly what, but we would like to have the students to participate in it. So make sure that if you're a student, a trainee in the center, that you want to volunteer to help organize the center together with, and then we can put the resources and the proper forum for that discussion. In a way, we can do that in a more planned and thoughtful way. Okay, so read the email again. And if you want to volunteer and be part of this effort in the future, please volunteer on Friday. Thank you.
Yes, I hope we have a meaningful discussion on Friday. There's another question from student David. Implicit bias is big in academia, and I've not seen any steps to tackle this from my personal experience. Universities should offer training to tackle this problem and case studies. A very good point. And we need to be good listeners and actively anti-racist and actively anti-discrimination. It's a comment from Harold Hastings.
Rita, can I follow up with this?
Yes.
So I have an extensive training about identifying implicit bias in academia by being an administrative fellow at UH. So one thing that all of us can be involved, can do is consider what constitutes excellence. I encourage to bring the idea of a rubric matrix when a group of people trying to rank what is a good candidate. I think in FIS program, I have trained the mentors. First, think about what are the qualities you think is actually exceed your expectation. And make sure that your own line of definition will not be the only line of definition for success. And I bring such idea into the APS Fellowship Committee because, you know, APS, American Physical Society, is not a very, it's trying to be inclusive. And then we wanted to do that by bringing a lot of best practices to fight biases into our society, into our daily activities. So I hope that one day we will be successful, and we are succeeding. Thank you.
Yes, absolutely.
Yeah, and hi, I'm Emil Kraus. I'm a grad student just listening in on these talks. Really like that this conversation is happening. And in terms of what you just said, Arpita, yeah, like to get at the more high school level students that, you know, drop out, those barriers. I've seen some of this, but universities started to rely less on the GRE and the subject GREs because they're major financial hurdles, especially at that stage. And there is a lot of research out there that they're not a good predictor for, you know, eventually, you know, they're not a good predictor for research success. Or I saw someone in the chat say a big problem is merit is so narrowly defined. And I really agree with that. Yeah, so we gotta, yeah, I don't know.
Yeah, absolutely. That's a really good point because I think there's a lot of drop off before we get to even the graduate school stage and beyond. Mm-hmm. So there has to be some efforts, systematic efforts to increase the inclusion starting from early on, I think, especially in STEM fields, because the drop off is already so large by the time we come to graduate school that it becomes very difficult. I'm going to copy down these links that people are sending, and maybe we can send an email around with some resources. I think we can all try to identify some institutions in our own localities that we can reach out to if we aren't already doing that. Some of us are involved in some outreach activities, but we can try to be more active.
I think that's a great idea, Rita. I think that FIS has done very successful outreach, and also we have assessment. So which means that a lot of our outreach activity is driven by hypothesis and also work with evaluators to conduct formative assessments. So we can change and then we can improve as we evolve, and we change with location and we change with school culture. So I think if PoLS, if any PIs will be interested in teaming up, you know, about reaching out to minority students, we'll be very happy to support you.
That's fantastic. Yeah, because I mean, again, in the DC area, the Maryland area, there's definitely many minority institutions that we can actively reach out to from my point of view, for example. And it would be very good to connect on that. Trying to see if there's any other specific questions. If any students want to, talk or say something, feel free to unmute yourself. Oh, that's a great idea, Marky, about saving the chat, because I was trying to copy things down. Okay, I think this is a good starting point for us to think about this. Spend some time reflecting today, maybe think about what specific steps one could take, small or big, and some small steps that could merge into bigger steps over time. And we can create a space where we share links and resources, ideas, specific ideas that could be implemented, both on an individual basis as well as sort of an institutional basis. I think that would be very good, to have some sort of a resource that one can turn to, a set of resources. Okay. Does anyone else want to say something today? Okay, well, thanks, Margaret and everybody, for the discussion. These are really important points, very relevant to students at all levels. And
Marty, you're going to send out a revised schedule for Friday, right? Where we'll have a follow-up to this discussion where people can use what they're reflecting on and try and plan for their action. Is that correct?
Correct. There will be a revised schedule with a link for Friday's meeting, and the discussion around planning for the next for a diversity meeting. And thanks to ARPITA for leading us this afternoon. And if our speakers are still here, I would say, I'm going to put the link again. If y'all still want to have speaker questions, if they're not already there, but you can go and talk with the speakers. And I think that's about it for today.
Yeah, thanks to all the speakers who spoke today, and thanks to all of you for participating as well. Yeah, look forward to seeing you on Thursday and Friday. And Friday, we will have further discussions. Thank you. Thanks, Margie.